This is SciFi Talks, where we talk about science and finance with investors, entrepreneurs, and academicians in life sciences. We invite guests, and for today's episode, we have our very special guest, Dr. Bikash Shakya. Dr. Shakya is a technology development scientist at Life Edit Therapeutics. Life Edit Therapeutics is a startup based in US, developing novel human therapeutics for genetic diseases, or simply the startup editing the life. Welcome, sir. Let's
Thank
start
you.
with a very general question. So, where is biotech engineering now? I mean, advancement-wise, the development-wise, what is the status of the biotech engineering now?
I think the biotech industry is booming quite a lot, especially in the field of human medicine or in the field of therapeutic diseases, therapeutic target for the diseases, and uh, the related to things related to the human biology, especially after the this success of the gene sequencing project for the human genome. A lot of resources have been focused on human biology and the disease targets. So I think biotech industry overall is going pretty good.
Uh, we understand there could be some confidentiality, but if you could tell us about the Life Edit Therapeutics where you work and your work there.
Life Edit is basically a subsidiary of a bigger company called Elevate Bio. Basically, Life Edit is morely focused on gene editing technologies. At Life Edit, we work on a form of CRISPR. We call it CRISPR 2.0. Basically, it's also called base editing. And we have a library of proprietary nucleases or base editors that were derived from agricultural biome. And Life Edit also works on or develops number of engineered nucleases, and we basically screen those nucleases to use them as a component of CRISPR as a base editor, and identify the one with the high editing efficiency and use those for targeting different disease. So we work on different therapeutic target as well to treat those disease using our base editors and our nucleases. And we have several partnership with different companies, and we work on different disease target for those companies.
What you have to do your role?
Basically, I'm in the technology development group. So my work will be especially getting those engineered nucleases, engineered base editors from our protein engineering team, and screen those in the mammalian cell line to identify their editing efficiency and identify the lead candidate, or it's a lead discovery, I would say, and then promote it as a like lead candidate and pass it to the other group downstream. And beside that, I also work on cell line development, where I use the CRISPR to generate the in vitro disease model, which can be used to test the efficacy of our gene editing products.
Uh, so, so what is the business model of this company? Like, is it like a CRO, the contract research organization?
No, it's basically a research-based organization. It's not CRO. We actually are part of the Elevate Bio, and all funding comes from the Elevate Bio, and we actually identify those nucleases, identify those base editors, and use them for the further applications. And regarding the other funding source, we partner with different companies, and we work for them. It's not a CRO, but it's a kind of partnership program. For example, we have recent partnership with Moderna. We have recent partnership with Novo Nordisk. So where we work on the particular therapeutic target or disease target of their interest and use our technology to target the disease that they are interested in.
think your job is also amazing. So I would like to go back to your journey. So how was your journey from research constrained environment in Nepal to the top research labs and then again to the impact making startup like this?
Yeah, so going to my background, I did masters in microbiology in Nepal from Tribhuvan University, and then I mean, at that time, when you do your masters, uh, your next step would be to find a good job for you. And at that time, I was like interested in like teaching positions in the medical college, and I was able to get that and started working in a national medical college, where I worked for almost 4.5 years. Uh, and in the beginning, I didn't have anything in mind regarding like doing the higher studies, doing PhD, those kind of thing. But as I started working there, I was also doing some kind of research, basic research, mostly focused on epidemiology. But after staying there for 4.5 years, I started feeling I should like focus my research, or I should learn about the molecular technology, molecular researches or studies at the molecular level. So I got interested in those things, and then thought of applying for PhDs abroad, PhD program abroad, and just started my application. And I got accepted by a university in the United States. Purdue University. So after coming to Purdue, I found the opportunity to explore more about the molecular techniques, using molecular techniques for infectious disease research. So if I compare the research environment in Nepal and the United States, it's definitely a huge difference. Like in Nepal, we didn't have much like funding sources. I had to do my researches using my own fund. Even my college or even my workplace didn't like sponsor any kind of thing or provided any fund. So, but coming to the United States, I found that there are a lot of funding opportunities. The universities help its faculty. There are a lot of outside funding agencies or government funding agencies as well. So it was totally a different experience coming from Nepal and looking at the research-friendly environment in the US universities.
So was it a plan, sir, to publish the papers while you were in Nepal? So was it a plan to get a PhD in US, or you had an interest to do research, so you did a research in Nepal?
Yeah, so after getting engaged in like master's thesis, I had some kind of passion for doing a research. So I wanted to somehow involve in researches, like despite of limited resources. So like I kind of carried out those resources. For example, like just doing simple study of like the intestinal parasites in the school, prevalence of those parasites in the school, or just looking at the hospital data and analyze the pattern of the disease and the patient in the hospital, and looking at the different microbiological aspects in different departments of the hospital. So those kind of research I carried out, and actually I published a lot as well, just based on that research. So I think I would say, besides teaching, I want to engage myself in some kind of research and learn more.
Okay, so there was a long gap between your master in Nepal and the PhD. So was it difficult to find PhD after that gap? So how did you
Uh,
manage?
Yeah, I don't think so. It was that difficult, but yeah, it took me almost like 1.5 years to 2 years to uh, get accepted. Besides United States, I applied in different other countries as well, like India, for scholarships, in the countries like Japan, in the countries like Germany. So the thing was, applications were competitive, and you want to get funded, and that was the main thing. So in the United States, when I applied, like I applied multiple universities and got accepted by Purdue University.
So you had a very excellent academic record, but still you chose industry, the startup in the end. So why industry in the end?
So yeah, when like I was doing my PhD, I didn't know the difference between academia and industry. While doing PhD, the thought was, or what I was thinking was, like I would be a professor. I would go to academia after my graduation. But in the United States, I found that, especially for the biology students, the process to get into the academia is not so straightforward, and not very easy as well. So it was actually difficult to get into the academia, and you have to do like postdocs and get more experience uh, in your research, in your field, before you apply for the academic position. So I joined postdoc, and as I was doing postdoc, I started to find the differences between working in industry and working in academia. And uh, for a number of reasons, I got more interested towards industry rather than academia. So basically, some of the factors were like in industry, usually it's fast-paced environment. It's a more dynamic environment in the industry as compared to academia, and there are more like funding opportunities in the industry as compared to academia. So these are some of the factors, and on the top of that, what I felt was maybe like looking at others who have joined industry and who have joined academia, I started feeling maybe there is more work-life balance in industry as compared to academia, and thus I decided to go to industry, like leaving my previous goal of becoming a professor.
So what's the long term plans, to stay in the industry forever, or you just want to go to the academia later in life?
I think I would stay in industry, and I'm enjoying the environment in the industry, working environment, work culture in the industry, and like type of science they do, using the advanced technology and the accessibility of resources. So based on those things, I think I would stay in the industry.
So my another question is, there is a fight like big, and some people love to work in a big industry, big pharma, but some small startups. So what is your choice for working? So what do you prefer for working?
Yeah, so when I applied for industry job, I applied both big industry and small industry, and there were a couple of like offer from the big industry as well. Um, but mostly from the small companies, and based on number of factors, I choose the startup companies, small companies. So I don't have like exact experience of the big industries, but what I have felt in the small industry is, uh, in the small industries you have more opportunities, especially more opportunities to grow, more opportunities to learn, and you have to wear a number of hats in the small industry that gives you exposure to like number of things or number of sectors that is good, and that provides you good learning experience in the small companies. And next thing is, in small companies, it's more dynamic environment, but some of the things that may like deter you from your good experience, or from what you want in the industry. Maybe like sometime the priorities change very fast in the small companies, small industry, as compared to the big industry. But I would say maybe big industry have more stability as compared to small industries.
Another advantage of working in a small startup is you can be a part of the growth of the. You can be like a founding team. I think that's
Yeah,
one advantage.
you can get involved in like from the start of the project, or
Yeah.
from very beginning in the project.
I think they you can develop a feeling like this is my own startup. I think that kind of feeling exists in the small startup. Like, what is your suggestion to someone like me who has a master degree, or maybe someone who has a PhD, and the guy or someone who wants to break into a biotech startup? What is your suggestion for them?
I would say find a, right company that match your skill, that match your interests, and that match your passion. That way you'll be able to impress the hiring team. So, and don't hesitate to reach out to people, network with the people in the industry where you want to get into, and that will make you visible. And Yeah, I think first thing is, especially with the startup or in the industry overall, I would say your skill and your interest should match with their job description, or what kind of people they are looking. They won't be just looking to fill a position, but they would be looking for a person who can lead their entire project. So those kind of people they want, who are ready to accept the challenges, who are ready to wear multiple hats and go above and beyond the like what they have been assigned, or they will be assigned. So yeah, those kind of mindset, if you have, and if you can develop yourself according to like their need, then definitely you have chance to get into the industry.
And let's talk about the gene editing. Like, it's very trending, it's in hype, but is it being applicable in the real world? Like the way it is making trends and hype, like is it being applicable, or is it just being limited to the R&D and in some part, but it is not making such an impact in the real world? So, what do you say about the gene editing?
Yeah, so the history of gene editing is like, especially the CRISPR, is not very old. So just in 2012, we got the first paper on CRISPR, and it's been just like 10 to 12 years only since we started to know about the CRISPR for the gene correction. And it got hyped, especially after the Nobel Prize that Jennifer Doudna and Mendel Carpenter received in 2020. And in terms of like the application, I think it's still like under progress, and there needs to be done a lot of things to make it applicable in the human. And a lot of researches are going at the basic research level, at the preclinical level, like very like encouraging results have been obtained in animal models, but we are yet to obtain like good result in from the clinical trials. But as like dozens of clinical trials are going on regarding the gene editing or use of CRISPR and its variations of the CRISPRs worldwide, I think we will soon see some encouraging result from the clinical trials as well. So the genetic medicine, I think, it's the future of the medicine, and I'm pretty hopeful about the good results from gene editing.
Okay, so I think when we talk about the gene editing itself is progressing. Like there were different approach of gene editing, like CRISPR was one, and again you said just like base editing is another approach, right? So how is it progressing? Like it doesn't stop somewhere because
Yeah. So that's a good question. So CRISPR usually it comes the use. How CRISPR works is usually it causes the double strand break in the genome, and that double strand break can result into a number of off targets, number of aberrations in the genome, and in order to overcome that thing, base editing came, and in the base editing there does not occur the genome editing, sorry, there does not occur the double strand break. It just does a nicking, that is a single strand break, and it changes the nucleotide from one nucleotide to another nucleotide, suppose from adenine to guanine, so A to G transition, so without the double strand break. So that now overcomes the bad or disadvantage of the double strand break. And but when you are doing the base editing, there is some chances of editing the undesired nucleotide as well in that window. So in order to overcome that, now there is another form of the gene editing, which is called prime editing, which is booming a lot. So in the prime editing, you can correct the gene by changing the nucleotide, or you can even do the insertion or deletion of the nucleotide sequences in the prime editing, and that overcomes some of the disadvantage of the base editing. And there is again another field of gene editing is booming, which also uses the CRISPR protein, that is called epigenome editing. And that epigenome editing use the dead nucleases. That means that does not cause any kind of break. So in this way, I mean, the CRISPR is going forward, and it is trying to improve itself so that we can have a genetic medicine that can be used without having much side effects, or much off targeting, or without creating much damage during the treatment.
I think it's the main challenge we find in the gene editing, the off target in the human genome, right?
Yeah, off target is one of the challenges, and besides off target, I would say the delivery, how you deliver those CRISPR components or gene editors into the human body. So nowadays, I mean, people are mostly focused on delivering through the virus, packing them in the AAV virus. And when you use AAV virus, there is the packaging limit of the AAV virus. So if you need to deliver bigger component, or if your design has a bigger component, then you can't really use AAV. So it has packaging limit, and now people are using the lipid nanoparticles, and lipid nanoparticles are becoming quite popular to deliver the gene editing components. And next challenge is the immunogenicity. So, for example, the Cas9 protein is derived from the bacteria. So, the bacteria that you are exposed to, or your body is exposed to. So, when you deliver that Cas9 or Cas protein into the human body, your body can have immune response. So, there are the challenges like off targeting, immunogenicity, delivery problem, and people are finding solutions to overcome this problem.
The case in the gene editing when you try to use gene editing as a therapeutics, but when we try to use the gene editing as a platform, to do other tests, or maybe to do other R&D activities. So I think there won't be a problem, right, to use the gene editing approach.
Yeah, there won't be problem. In the R&D or in the development stage, we usually don't care much about this type of thing. We just care about like what is the editing efficiency, how much editing efficiency it is giving, and then you go into the mouse model or small animal model, and then you start to face the problems like delivery. You start to face the problem like the immunogenicity, and people try to find the ways to overcome this. And like if you are not using the CRISPR technology in the human, I mean CRISPR, the application of CRISPR or genome editing is not only in the like human disease. It can be used to improve the like productivity of the plants. It can be used in the diagnosis of disease. I think there was a technique to detect the COVID using the CRISPR technology as well. So besides human disease, besides human targets, there are a lot of other applications of the CRISPR as well.
We don't understand the proper biology of the things, and one gene we are trying to knock out might be useful for certain purposes, and we might know just later on. So sometimes do you get afraid because of this kind of things, because we don't know the proper biology and we are just using the biology we have been knowing, but in the future we don't know, because it might have certain impact later on. So sometimes, so does that create a fear in you, on you, or not?
Yeah, definitely. I mean, there can be that kind of fear as well, but that's why there are technologies to find out like what will be the other impacts of changing this gene or editing this gene. Usually, if there is more benefit than the amount of damage it does, usually you promote that or you accept that. Like there are a lot of diseases, especially say a lot of rare diseases, where like kids die at very early age because of the mutation in the gene, because of some kind of changes in the gene. Then at that point, what we should think is if the gene editing can like lengthen or provide the kids a little bit of longevity, or it allows the kid to survive longer, then definitely that gene editing is a boon for that kid and should be applied. And not only like rare diseases, the gene editing is being used for a lot of other common diseases as well, or being studied for a lot of common diseases as well. And in that matter also, like if you find something very good in correcting that disease, definitely you should be like concerned about what can be the bad impact or what can be the side effect of those things. And if you think if those side effects can be minimized, and if you know those side effects and some way if you can reduce that damage or harm, then definitely then that gene editing can be used for treating those diseases. But like we don't know everything. Sometime you see something and that may not be the only truth. So there can be a lot of hidden things, and that's why we need like continuous research and use of technology to find out those things, the hidden things.
Yeah, and this is biology.
Yeah.
Several unknown unknowns, known unknowns, and many secrets are there still to be revealed. So coming from the resource constraints and a no top research experience, maybe just few research experience, does it make our research career and competition later more challenging? Or maybe like like you can just say relating with your the problems that you faced in your PhD, because the people we have to work with the people from the diverse background, from the diverse nationalities. So did you face some of the problems while working in the US?
Yeah, definitely. coming from the research constraint environment, we felt ourselves less competitive. So at the time when the world was like already doing advanced molecular research, like in our country, we were trying to do PCR or just to run a gel, make a gel, or we're trying to learn how to operate the PCR machine, and we had like only one PCR machine in the entire department. So we came from those kind of situation, and coming from those situation, definitely I found myself very behind, behind after joining the lab in the United States, because I was trying to learn very basic technique. So skill wise, I was very behind. Maybe I had good theoretical knowledge, but the skill wise, technique wise, I felt myself behind, and it took some time to learn those things, and then like to level myself to the other colleagues in the lab. As the technology is developing in such a fast pace, there is a need that we should prepare ourselves to keep up with those pace. And I think in Nepal nowadays, it has improved a lot in the sector of molecular research as well, in the biotech sector as well. But still, we may be far behind. So, like if you think about the current hot technologies for like disease treatment research, for example, gene therapy or gene editing using CRISPR. So I don't know how much people are thinking, or people are trying to, or putting their effort to like introduce this thing in our own country. Not at the like higher level, but at least at the basic level. Are they teaching about this type of new technology to the students in our country or not? So that is a question. So here now people have started to learn and use their skill to do the gene editing or CRISPR from their undergrad level. And in our country, are we or have we started teaching or educating our students about these new technologies or advanced technologies? So that is a question. So we should definitely should be updated, and at least theoretically, and if possible practically or technology wise, to our best, based on the available resources, we should try to introduce this thing in our curriculum in the country.
Yeah, I agree. You invested a lot of time in the research activities, like even in the Stanford University you were, I think, which could have
Yeah.
generated a few startups too. So, is it possible to create a startup from academicians working in a foreign research labs? Because we expect when we are in Nepal, we expect bigger things like, oh, okay, I will go to the foreign lab and I will start my one startup. So, is it possible, or are there many barriers so we cannot get out of that?
So I think it depends on the type of research or the field of research that you do. Like, can it be translated into a startup or not? So basically, when you are doing a basic science research, it may be a little bit difficult to do that, because all the research don't translate into startup. But having said that, the other thing is the startup usually, most of the startups, they are kind of spin out from the academic lab. So in the academic lab, suppose if they are working on certain technology and they feel the technology works, then they start a company just based on that technology. Or if in a academic lab, if they are working on a particular disease and they are doing some kind of let's say preclinical research, and they got some interesting data in animals or mice, then they can go and start a startup that does like a clinical trial or clinical studies for those kind of disease. So what I mean to say is startups um, come or spin got or come out of the academic lab, but your question is like the people working in the foreign labs, can they start their startup in Nepal? Is that your question?
Uh, you study in Nepal or even abroad. So is it possible to do for
Yeah, I think
academicians? Yeah.
yeah. So the academician, they can definitely start the startup, and they have done that as well. So a lot of PIs or professors, they are involved in number of startups, and their technology, their studies have generated number of startups as well. So that is definitely possible, and as I said before, it depends on what kind of research or what kind of field they are working on.
But is it possible to bring that technology that you did there and bring it to Nepal? I think they are.
Uh, so the technology wise, I think suppose in my postdoc, I learned CRISPR. I used CRISPR to modify the hematopoietic stem cell, so that we can get the red blood cells that is devoid of a particular protein, and studied like if that red blood cell is that susceptible to the malaria infection or not. So technology wise, I learned how to grow the hematopoietic cell, how to differentiate them into the red blood cell, and how to modify the red blood cell, or how to modify the hematopoietic stem cell. Similarly, I did some CRISPR in malaria parasite as well. So this technology that I learned definitely can be brought to our country if we are doing some work that utilize those kind of technology. And definitely, if there is a suppose, let's imagine a company is getting started in Nepal. A group of people are starting a company in Nepal, and they need this type of technology, or they need someone who are expert in this type of technology. Definitely I can be helpful. So if even if I'm not directly involved in the company, but I can serve as advisor or scientific advisor to help them establish those technology in that company, or not only in the company, but also in the academic universities or colleges as well.
Let's just talk about Nepal. Even in Nepal, when you see the condition of biotechnology, like you see many research institutes or the non-profit organizations or the NGOs doing the research, but very few biotech startups. So, what do you think? Like, what kind of biotech startups could be imagined in the countries like Nepal?
I think based on the scenario of Nepal, based on the richness in the biodiversity, based on most of the people, a lot of people involved in agriculture. So I would say the biotech, like based on this type of sector, for promoting or for utilizing the biodiversity, for improving the agricultural product, or for improving the productivity of our lands. So those kind of biotechs, I think, would be more suitable for the country like Nepal, rather than the biotech that are focused on developing the therapeutics for the disease. And definitely talking about the biodiversity, there can be a lot of medicinal plants. So if any biotech can be imagined to use the medicinal plants of our country for treating the disease, and I think that is also possible.
Like, can we use gene editing technology to it in the biodiversity, the startup?
Uh, So it depends. I think if there is a need, for example, we identify, suppose a plant, a medicinal plant uh, that can be, or whose extract or whose product can be used for like a component of some kind of medicine for treating the disease like cancer or anything. And then suppose if we are finding trouble growing that plant in the environment we want, or maybe suppose they grow in the higher altitude and we want to grow them in other environment or in the lab or in kind of other type of niche, and maybe we need to use some gene editing technology to make it able to grow in such kind of environment. Or let's say if you identify the plant, but the problem is you have some kind of the plant gets attacked by some kind of disease, and you need to control that disease to increase the productivity of that plant, that medicinal plant, or to preserve the component that you wanted from that plant. Then definitely gene editing can be used to make that plant resistant to that particular disease that can be imagined. So definitely, I think there is the use of gene editing.
Like you said, gene editing and therapeutics in countries like Nepal is really can be a problem as a challenge. So, what do you think about CRO model in countries like Nepal? If we are doing gene editing, because we have expertise, I think we don't lack expertise. So, if we get backed by the expertise, so is it possible? What do you think about this model?
Yeah, I think that is possible because we see like a lot of things that are being done or that are undergoing in the United States. They are like handled by CROs in other countries, especially China. The labor being cheap. Suppose if you want to make certain plasmid, or if you want to make certain virus or that are used for the research, if you want to make some kind of protein or antibody, uh, the companies in the United States outsource from China. So they are being made in China and sent to the United States. So if we have resources, if we have labs, if we have companies that are capable of doing that in Nepal, or if we can establish those kind of CROs or CDMOs in Nepal, then definitely uh, like the companies outside Nepal can have the services from Nepal based on the quality, based on the price. That is possible, and for this, I think the government should make policy to encourage the establishment of this kind of companies in Nepal.
How often do you use the bioinformatics? Because it's also very trending a lot nowadays. The students are thinking of starting their career in the bioinformatics and in the data science. So, what do you think about these disciplines, the bioinformatics or the data science and the machine learning, AI, these kind of things? Like you are working in a startup, so how often do you use this technology? Are there already other people doing these things? So, what do you think about this discipline or the domain?
Yeah, I think bioinformatics is the integral part of biological studies or biological science nowadays. And talking about like not only my company or our company, but in all other companies as well, there is like separate bioinformatic group, and especially at Life Edit, like a lot of things are based on the bioinformatics because we are trying to mine the nucleases or that nucleus protein from the microorganisms. And for that purpose, the bioinformatic is required, especially for analyzing the gene sequences. Similarly, as I told before, like we do the protein engineering as well. So engineering of the protein sequences to make that protein more effective in the CRISPR technology. So for that also, the groups in our in our company are using the machine learning and bioinformatics like in wide scale, and that I think applies to other companies as well. So bioinformatics is like integral part of the biological science.
I think we are in the end. So, in the end, I would like to ask the questions like what suggestions do you want to give to the young students who want to do great research in great labs like you did? Maybe specifically, what skills should they try to develop while they are in Nepal or in any research constrained environment? So, what skills are the most important?
Yeah, so if you want to get into the good lab and do further studies, definitely like I would encourage them to prepare accordingly. Like what you want to do, what kind of research you want to do, or where do you want to go. Look at the expectation, look at the need for those opportunities, and try to develop yourself accordingly. For example, if you are applying for the PhD programs abroad, definitely the research experience can help you a lot. So keep on doing any kind of research that is possible in the country. Try to publish papers in different journals, national, international journals, or local journals, at least to prove yourself that you can write something about research, you can analyze data. So those kind of skill set you can develop while staying in Nepal. And I would encourage communicating with the people abroad, communicating with the professors. You may not get the reply from all the people you contact, but never like stop communicating, never stop writing emails to them, showing your desire, showing your interest, and show yourself that you are passionate about the things that you want to do, and always keep open mind, uh, and think big. I would say, yeah.
Yeah, I think big is a great suggestion. Also, and how can someone reach out to you?
Yeah, I can be reached out easily through LinkedIn. So if you need any kind of help, if you need any kind of suggestions, guidance, or mentorship, uh, you just like send me a message in my LinkedIn profile. I will try to reply to it as soon as possible. Yeah.
In the end, as a conclusion, I can say that gene editing is a great technology. It's useful for different purposes. Even in Nepal, it can be applied for the, I think, for the biodiversity. Like we would like to conclude by saying this, and also you gave a very important suggestions to our young students, and which is very useful. Thank you, sir. Thank you so much, and thank you so much for joining us.
Yeah, thank you so much for having me. Thank you for this opportunity to share my experience and share my thoughts with you.